Welcome to today's lesson on the big, bad, monstrous, nightmare-inducing Krebs Cycle. Just hearing the term Krebs Cycle used to send chills down my spine as a high school student and as a college student. So hopefully I can break it down for you so it is not so terrifying and so that it makes sense. And at the end of this lesson, at the end of this unit, you can brag to everybody about how you are a Krebs Cycle expert. So we're going to pick up where we left off last time with glycolysis. And during glycolysis, we broke glucose to make 2 pyruvates. And we made 2 ATP. And 2 ATP is not really all that much. So we're going to keep breaking down the pyruvates and trying to get some more energy out of it. But before we look at the details, let's look at the big picture. So the Krebs cycle and glycolysis, they're all part of cellular respiration. And cellular respiration has one major goal. It's to take sugar and to get some energy out of it in the form of ATP, to break that sugar and get ATP, because ATP allows your cells to do work and it allows you to do work so that you can do the things that you love, like read books, talk to friends, watch movies, all those things that you like to do. You need ATP for it. In this process, oxygen is required to help break that sugar apart, and carbon dioxide is produced as a byproduct as well as water. So we're going to take a look at the steps of cellular respiration. And last time, glycolysis was our topic, and we took glucose, a 6-carbon molecule, and we broke it into 2 pyruvates, which are 2 3-carbon molecules. In the process, we ended up with 2 ATP and some other stuff, and today we're going to look at the Krebs cycle, which is right here. And one thing you can notice here is glycolysis doesn't happen inside of the mitochondrion. It happens in the cytoplasm, if you recall. Today, we're going to enter the mitochondrion for the Krebs cycle, and we're going to be looking at processes that happen in here. But before we do that, just to look at glycolysis, this was the diagram from last time, right? Starting with glucose, and it finished with 2 pyruvates and some other important stuff, including 2 ATP and 2 NADH, which are these high energy molecules that we are saving up and we're going to use when we get to the electron transport chain, the final stage of cellular respiration. So the second stage of aerobic respiration, as I mentioned, happens in the mitochondria. Specifically, it happens in the mitochondrial matrix, which is this inside part of the mitochondrion. Now, I have some bad news for you. You think the next step is the Krebs cycle, but before we get to the Krebs cycle, we actually have to do something else because the pyruvate we made during glycolysis is not ready for the Krebs cycle. We have to do something to it first, and that something is the formation. Instead of starting with pyruvate, we're going to make something known as acetyl-CoA, and that's what's actually going to go into the Krebs cycle. All right, but I'm going to break it down for you one step at a time. So here is our big scary graphic showing you acetyl-CoA formation and the Krebs cycle, but don't worry, we're going to look at it one step at a time and take it slowly. So we start with the pyruvate from glycolysis, right? And what we're going to do to it is something known as pyruvate oxidation. We are going to change the pyruvate into something known as acetyl-CoA, which is what we need to start the Krebs cycle. And how we do that is we're going to break a carbon off of the pyruvate, and that carbon is going to go somewhere. Where is it going to go? It's going to form into carbon dioxide, and carbon dioxide is going to leave. All right, if it's a human we're talking about, we're going to exhale that CO2. Okay. The other thing that happens in this process as we break off one of these carbons is NAD plus becomes reduced and we form NADH. And that NADH is one of those useful high energy electron carriers that we are going to save for the electron transport chain at the end of cellular respiration. So that's something cool that we made. All right. So pyruvate, we break off a carbon and we end up with acetyl-CoA. So that is step 1. In the next step, we are going to enter the Krebs cycle. And I should tell you that the Krebs cycle has another name, and that name is the citric acid cycle because of the formation of something known as citrate or citric acid, which happens in this step. So what we're going to do here is we are going to combine the acetyl-CoA with something known as oxaloacetate. Oxaloacetate, super fancy name. So we're going to combine acetyl-CoA with oxaloacetate, which isn't drawn here for you, but this is oxaloacetate. That's what it looks like. And the 4-carbon molecule, because oxaloacetate has 4 carbons and acetyl-CoA has 2, together they're going to form a 6-carbon molecule. I'll prove it to you. 1, 2, 3, 4, 5, 6 carbon. And that is known as citrate or citric acid. It's the same thing that you find inside of lemonade, so it's not something that's super, super weird. In this process, coenzyme A is also regenerated, which is something we used earlier. So that is step 2. We took acetyl-CoA and oxaloacetate, which is pictured here, and we combined them to form something known as citric acid or citrate. Okay, in step 3, what's really going to happen here is we're actually going to start breaking off the carbons from citrate. And we're going to break off those carbons and we're going to be releasing them as carbon dioxide. And we're going to be making things in the process. We're going to be reducing NAD plus and start making some NADH molecules. And we're also going to make some ATP. So here we're going to take it one step at a time. So in step 3, we broke off 1 carbon and we released some CO2, and we also made 1 NADH, which happened right here, right, when the NAD plus combines with released electrons and a hydrogen ion, right, it became reduced into NADH, right. In step 4, we're just doing really the same thing. We're breaking off another carbon, which leaves us carbon dioxide, and we're making another NADH. So at this point, we've released 3 molecules of carbon dioxide, 1 here, 1 here, and 1 here. And remember, this process is actually going to run twice because we have 2 pyruvates, so we're going to do the Krebs cycle 2 times. Okay, so what happens next? In step 5, here we are forming 1 ATP. And again, we're going to run this process twice, so we're going to form really 2 ATP. In the next step, in step 6, we are actually going to make we're going to take the coenzyme FAD, combine it with electrons and hydrogen ions, and we're going to form FADH2, right? And that was one of those electron carrier molecules that we discussed in the previous lesson, something really useful for the electron transport chain too. So we took FAD and we made it into FADH2, which is the reduced version. In step 7, we're going to make another NADH over here. All right, so basically we're accumulating NADH, ATP, and FADH2. And then in step 8, we're going to regenerate that oxaloacetate, that fancy 4-carbon molecule, so that we can keep running the Krebs cycle over and over and over again. So that, in a nutshell, is how the Krebs cycle works. But here is a nice summary for you. It takes 2 turns of the Krebs cycle to break down a whole glucose molecule because the Krebs cycle uses 2 pyruvates from 1 glucose molecule. So here is what is produced after the Krebs cycle is done. We end up with 6 carbon dioxides, right? 3 carbon dioxides at each turn, 2 ATP, which is like pretty disappointing. Like, we did all this work just for 2 ATP. Like, that's like nothing. Can't believe you had such a crazy difficult cycle to make 2 measly little ATP. But here's the good news. We made 10 high energy molecules. We made 8 NADH and we made 2 FADH2s. And these are going to be super useful in the last stage of cellular respiration, which is the electron transport chain, which you will learn about next time. All right, and that's what it says here. The high energy molecules, we're also going to use the high energy molecules from glycolysis, which is another 2 NADH, and all of these, so the 8 NADH, the 2 FADH2, and the 2 NADH are all going to go to the electron transport chain. And here's a nice summary of all these things that are happening, right? We have 2 pyruvates. We're going to form acetyl-CoA. We're going to go to the Krebs cycle, and out come 2 ATPs, 6 carbon dioxides, 8 NADHs, and 2 FADH2s. And this all took place here in the mitochondrion. Now, I don't have a cool video for you, unfortunately, about the Krebs cycle. I have nothing very fun and interesting to watch, but I have this cute little comic showing you how a mitochondrion and a chloroplast are holding hands, and the 2 processes, cellular respiration and photosynthesis, are interrelated in this big, happy cycle. So I really hope that was helpful. Thank you so much for listening and watching.